Okay, this video is about the cell cycle. You are about 100 trillion cells, and there are 10 to 20 times that number of bacteria inside of you. This video is going to be all about what happens in the life of a cell and what goes on at different times. So this diagram shows the phases of the cell cycle. There are two basic phases of the cell cycle. There's the mitotic phase, which is when the cell divides, and the other phase is interphase, which is the time between cell divisions. So cells need to divide for three basic reasons in multicellular organisms. They need to divide for growth, repair of damaged tissues if you get a cut, but also cells just die of old age. So to replace dead cells that are just too old and have died of, I guess, natural causes. So interphase includes G1, S, and G2. 90% of the time that a cell is alive, it's in interphase. The first phase is called gap 1, and during gap 1, the cell grows and RNA and proteins are synthesized. And which proteins are synthesized depends on the type of the cell. So going back to first semester, if we were talking about the beta cells in the pancreas, we know they make insulin. So they'd be making insulin during G1. During the S phase, the DNA of the cell would be replicating, making an exact copy. And you really never have a change in chromosome number in mitotic cell division, but of course you would have double the DNA because you've gone from single-stranded chromosomes right here to double-stranded chromosomes. So even though there are two strands in this chromosome, it's still one chromosome. One half of that chromosome is referred to as a sister chromatid, and the center part here is referred to as the centromere. It is an unreplicated part that's in the middle of the chromosome, and it's important to keep the two halves of the sister chromatids together until right before you divide them, because that way you make sure you get one of every chromosome in there. And if you just divide chromosomes in, like the number of chromosomes in half, you might end up with duplicates of some chromosomes and other chromosomes missing. So in this way, you have your sister chromatids, this side is genetically identical to that side, so that when you divide them, when you end up with each new cell gets one of these sister chromatids. Well, they're not chromatids at that point, they're separated into single-stranded chromosomes. So the S phase, by the way, is referred to the synthesis phase, and what is being synthesized is DNA. So here we have G1 and the S phase. So this is an electron micrograph, an actual photograph of a double-stranded chromosome that is after DNA replication. And these two sister chromatids are genetically identical. So they're really sort of more like, instead of sister chromatids, think of them as genetically identical twin chromatids. And there is our centromere. Be really careful, because when you're counting chromosomes, it's very easy to think that this might be two chromosomes, but it's actually one double-stranded chromosome. So count the centromeres, and that is the number of chromosomes that you have. The G2 phase stands for gap 2. And during this phase, you basically have the exact same thing going on as you would during the G1 phase. You have more RNAs and proteins synthesized. So again, if we were pretending that this was a beta pancreatic cell, it'd be making more insulin during that phase. It's just a shorter phase than G1. And then the final phase that we need to talk about is called the mitotic phase. And mitosis is actually technically just the division of the nucleus, and cytokinesis is division of the cytoplasm and the rest of the organelles. And sometimes people will just refer to, cell division as mitosis. It's not technically correct. They're really using the word mitosis when they mean mitotic cell division. Now, we've talked about chromatids and centromeres and chromosomes, but there's a new C word here that we need to talk about called chromatin. Chromatin is a thready-like material It's another shape for DNA. So sometimes DNA is in the shape of the chromosomes, and other times it is thread-like. And the only time it's in the chromosome shape that you're used to seeing photographs or diagrams of is during the mitotic phase. During the rest of the time, it's actually uncoiled in chromatin, and it's actually easier to make copies of it and do other things with it and get the information to make proteins, because DNA is ultimately recipes for making proteins. So the chromosome shape is really good for dividing DNA equally and making sure that the right DNA gets into the new cells, but chromatin is easier for the other uses of DNA, which is to make proteins. Making the protein synthesis process will be in a different video. So not shown on this diagram is another phase called G0. Don't confuse it with go, because it's not go. It's actually kind of ironic that it looks like it says go, because it's sort of the opposite of go. The G0 phase is a phase that cells get into when they get stuck in the G1 phase for so long and they do not continue and divide, and so they're said to be in the G0 phase. Some common cells that are in the G0 phase are a lot of nerve cells and brain cells that don't divide as often as other cells. Skin cells divide very, very often because they need to be replaced because they're exposed to the elements and they die very quickly. Adult leaf cells are another example of cells that are in the G0 phase. If you tear a leaf on a tree and come back months later, it's still torn. It doesn't repair itself. What determines whether the cell gets stuck in the G0 phase or continues into the regular cell cycle is the buildup of a particular kind of protein called a cyclin. They are trigger proteins, and they build up right in the G1 phase and they send them into the S phase. And once a cell's in the S phase, the rest of the phases will continue. So there's sort of a stopping point towards the end of G1. And cancer is actually when cells are dividing too rapidly and spreading when they're not supposed to be dividing. So it's sort of almost like the opposite of the G0 phase in a way, because you have too much cell division. In G0, you do not have cell division. The difference is when cells are in the G0 phase, they're supposed to be in the G0 phase. So this next sort of half of the video about cancer really is more to prepare you for a project that you have coming up rather than the test. There are not a lot of test questions on cancer and its relationship to the cell cycle, but it will definitely help you for your group project later this quarter. So another name for cancer is a malignant neoplasm. We wanted to include that because you come across it a lot when you're doing research for your project. And so if you see that and you're wondering what it is, it's just a fancier way, a more scientific way of referring to cancer. So cancer is really a collection of related diseases, but their only similarity really is that they have uncontrolled regulation of the cell cycle. Cells are dividing without stopping and spreading to unique or other tissues, which is called metastasizing. Cancer can start anywhere in the body. It can be a solid tumor like breast or colon cancer, or it can be cancer of the blood like leukemia. So normally cells divide, and when they get old or damaged, they die. So sometimes in cancer, cells are dividing when they're not supposed to be, and sometimes in cancer, they're not dying and they're surviving when they should not survive. So there are three important types of genes that help regulate the cell cycle, and they are called tumor suppressor genes, proto-oncogenes, and DNA repair genes. And I'm going to get into a little bit of detail, but not too much, on each of these three types of genes and give you a good simile to kind of help keep them straight, because it's really pretty easy to get tumor suppressor genes and proto-oncogenes confused. It's kind of hard to tell the difference. So the first one I'm going to talk about is a tumor suppressor gene. So normally, tumor suppressor genes code for proteins that stop a cell from dividing when it's not supposed to. So it's like a stop signal to the cell. So if you look at the diagram here, in the normal protein, or when the gene is not mutated, you get a protein that keeps the cell division under control, and so you have a normal number of cells. If you have a mutation in in a tumor suppressor gene, the protein that it codes for is non-functional, and cells divide when they're not supposed to. So it's like the brakes in your car being defective, so you can't stop. So similarly, the cells keep dividing and they can't stop dividing. In many tumors, you get a tumor suppressor gene that's been inactivated or mutated, and it doesn't function and doesn't stop cells from dividing. Proto-oncogenes are genes that regulate cell division and cell death. Apoptosis is programmed cell death. It's cells dying when they're supposed to die. So apoptosis is actually a good kind of cell death. So a proto-oncogene becomes an oncogene when it gets a mutation called a gain-of-function mutation, and it's on all the time. And in this case, cells divide when they shouldn't, and they lead to cancer. This is sort of like if the accelerator in your car was stuck in the on position, and so it just kept accelerating and accelerating without, being able to stop. The third kind of gene that I want to talk about is called a DNA repair gene. So DNA repair genes find mistakes in DNA and repair them. And they're functioning throughout the cell cycle, but mostly are active in G2 phase because that's right after DNA replication. And so that's when they're most likely to find mistakes, because mistakes happen during DNA replication. And you want them to happen right before the M phase, because once they start dividing, they're going to be passed on. So if there's a mutation in a DNA repair gene, mistakes can't be fixed. So typically, in order for cancer to occur, you have to have a mutation in either a tumor suppressor gene or a proto-oncogene and a DNA repair gene, because often if you have mutations in the previous type of genes, if the DNA repair genes are functioning, they'll find those mistakes and fix them before cancer happens. So you're going to be given an exit card in your note packet, and you're going to do these, and you're going to go over them in class.